Hey guys, so in this video we're going to be talking about chapter 15, the innate immunity, and in the following chapter we're going to be talking about the second half of the immune system, which is the adaptive immunity. So let's go ahead and get started. So the overview of the immune system is that it has two different types. So we have the innate and we have the adaptive. The innate is non-adaptive and it is the immediate response to a initial infection, and it's not specific at all. It's kind of like the more general all-purpose immune response that we have. And then over time we develop something called adaptive immunity, and that, like I said, it takes time to develop. It is very, very specific. So whenever you have a foreign pathogen inside of you, what will happen is that your adaptive immunity will then take over, and it can act actually faster to fight off the pathogen again that you've been exposed to. And that's where we get like our antibodies and everything, but that will be further discussed in chapter 16. So with the innate immunity, it is comprised of white blood cells. So all your immune cells, they come from bone marrow. And then from the bone marrow, what will happen is that it'll differentiate according to whatever pathway. So as you see that, depending on if it becomes a myeloid or if it's a lymph node, will determine its pathway, its cell fate. So these are the main ones that I do want you to know. I know there's some other ones on here that we won't really talk too much about, but I do want you to know like what each role is, and then we're going to discuss that too. So if you are a polymorphonuclear leukocyte, you are eosinophil, a basophil, a neutrophil, or a mast cell. And then if you're a monocytes, they differentiate into macrophages and dendritic cells. And then we have lymphocytes, which are the B cells, T cells, and natural killer cells. We'll talk more in detail about those in chapter 16, but I do want you to be familiar with them. But let's go ahead and talk more about the leukocytes. So we have the neutrophils, basophils, and eosinophils, and mast cells. So neutrophils are almost all of your white blood cell in your blood, and their main purpose is to go around phagocytosing microbes. That is their main goal in life, and that is all they do. They do that all day, every day, until they die. And then basophils and eosinophils, they don't go around phagocytosing microbes, but what they do is that they will release toxins that are toxins to the environment to where the microbe is, and then based on that, it will cause the microbe to disintegrate in a way, I guess. And then also, based on the toxins that they actually secrete to their environment, they also make blood vessels a lot more permeable, meaning that if we have an active infection, you want things to be able to travel more quickly in your bloodstream in order for all of your immune cells to get to the area. So the more permeable your blood cell is, the quicker your immune cells can get to the infected area and then reduce the dissemination of a infection. And then mast cells, what they are is that they are comprised of histamine and then heparin. These are primarily in the mucosa and connective tissue, and they don't enter bloodstream at all. So these guys are the ones that will cause like allergic reactions. So like if you have like an itchy nose and whatnot, it's probably because of your mast cells, because they are very, very rich in histamines. So histamine is why you get like an itchy nose and like a lot of boogies. But it's good. Okay. So these are just pretty pictures of a neutrophil and eosinophil. I'm not going to expect you to know, be able to identify the difference, but whenever like I say multinucleated, it's because they have a multi-lobed nucleus where it's not just one solid dot. That's what it means. Okay. So now let's talk more about other cells. So we have monocytes, macrophages, and dendritic cells. So monocytes, they are constantly in the blood, and they will migrate to tissues where they will further differentiate into a macrophage or to a dendritic cell. So if they differentiate into a macrophage, these guys are everywhere throughout your body, and they're the ones that are most likely to come in contact with the pathogen first, on top of antibodies too, if you've already been exposed to the microbe. But these guys, for the innate immune system, these are normally like your first responders. They're first on site, and they have two main functions. The first one is to go around presenting pathogens, or like presenting antigens to T cells, or they can actually directly kill depending on the pathogen that they come in contact with. So they can either flag down other cells to be like, hey, like you need to come kill this guy, or they can actually directly kill it, just depending on the microbe that it is in its midst. And then we have dendritic cells if a monocyte differentiates into a dendritic cell. These guys, they are solely located in the spleen and in the lymph nodes. What they do is very similar to macrophages, sort of. They do go around presenting antigens to T cells. So they do kind of the same function as macrophages, but they definitely differ in their structure. So if you see like the top figure is a macrophage, as you can see, it's very like globular, very big, very bulky. Whereas a dendrite, that guy, he's very spiky. So they definitely have different structures, but almost similar functions, but they also have different locations. So the only way that they kind of are similar is the fact that they do go around presenting antigens to T cells. But dendritic cells do have the ability of phagocytosing bacteria as well, but they almost always do presentation. So those are monocytes, those are macrophages and dendritic cells. And then here is the general pathway of phagocytosis. So like we have a bacterium, and then what it does is that we will go around engulfing it, and then what will happen is that after we completely engulfed it and everything, we'll form a phagolysosome. So a phagolysosome happens whenever that the bacterium and lysosomes come together, there's fusion, and then we have a phagolysosome after the fusion occurs. And then whenever that happens, the pathogen gets chopped up and then discarded. So that's the general picture. There is also a lot more that does happen, but I do just want you to know the general basis of phagocytosis. Okie dokie. So white blood cells. So I'm not going to expect you to know like the actual pathway of how they differentiate, but I want you to definitely know, why is that important? Why is it important that y'all know white blood cell differentiation? Because based on what elevation of white blood cell type that you have in your patient can help you determine actually what kind of infection that they have. So that's the importance of white blood cell differentiation. So if your patient has an increase of total white blood cell count in general, they might just be having a general allergy or infection, like a very basic one, nothing too serious. But then if they have elevated neutrophils, that can indicate a bacterial infection. If they have elevated lymphocytes, that can indicate a viral infection. And then if they have elevated eosinophils, that can indicate parasitic infection. So it is important why you do know this, and this will definitely translate into your nursing careers or your medical careers, whatever it may be. So I definitely do want you to know, why do we need to know this, if that makes sense. Okay. So this is just another table. I'm not going to expect you to know the normal values. You'll get that all in nursing school. But like I said, I do want you to be able to tell me the importance of white blood cell differentiation in the context of nursing. Okay. So with that, let's go ahead and go into the actual immune system, so that you can kind of break it down sort of into like 2.5 parts. So we have the primary lymph node organs, the secondary lymph node organs, and then the lymphocytes. So lymphocytes are the cell types. But the primary lymph node organ, that is where the immature organs will mature. So the two main organs is the bone marrow. So bone marrow is inside your bones, and that is where B cells are comprised. And then we have our thymus, which is the organ that's located in your neck throat region, and that's where T cells are. And then from there, we have the secondary lymph node organs. So this is where lymphocytes, they will actually encounter true antigens. So this is where they'll kind of encounter the bad guys, if you want to say it like that. So that is where we have our lymph nodes. So our biggest lymph nodes are located in our neck region, armpit, and groin region. Those are our big ones. Your armpits and your groin region are the biggest. And then from there, it does branch out through your entire body. And then we have our spleen, and then we have pyre patches, and then on top of our tonsils and also our appendix. Some people don't have the appendix anymore, which is perfectly fine. In my opinion, it's kind of, I don't want to say it's a useless organ because it did have a purpose, but I think evolutionary speaking, it is becoming rather redundant for our body, I guess we can say, but it does play a minor role. And then we have our actual two lymphocytes, so our two cell types, so that's B cells and T cells. B cells specifically will produce antibodies, and we'll go more into that in chapter 16. And then T cells, these are the modulators, so these are like the referees. These are kind of like the guys that you see at the airport directing the airplanes. So these are those guys with the wands on the runway. I don't know their real names, and that's really bad. But those are the guys. They're the ones who tell the cells, like, this is where you need to go, this is what you need to produce. So they help modulate our adaptive immunity, and we'll go definitely more into detail in chapter 16. Okay, so this is the overview of everything. So we're primarily going to be focusing on our what's it called? The bone marrow. We'll be focusing that on later, and then definitely the pyre patches we're actually going to mention in the next few slides. So this is kind of just the basic anatomy. You might have already seen this already. Okay, so the actual first physical barrier of our innate immune system is our skin. Our skin is probably, to me, one of the most important organs. So it is a protective shield. It is comprised of keratin, which is made by keratinocytes. And then also we have something called sebum. Sebum is that oily substance on your skin. So sebum is actually something, some people produce more, some people produce less. It kind of just depends on the person. And then a lot of times people who have a lot of sebum or a lot of oil production tend to have more acne sometimes, depending on the person. But sebum is a good thing. And then also from that, we are slightly acidic on our surface, which does help inhibit microbial growth for us, so that's good. And then also because of that, and because that we do have some microbial growth on there, we do have competition between species. So with the fact that the bacterium, or not just bacteria, but microbes themselves, they are competing for land, essentially. It helps minimize what can be colonized. So that's really good. And then also the fact that we're constantly sloughing off our epithelial layer, we always get a fresh new skin. So we always get a fresh new coat on ourselves. So that's also really good for us. And then also we have something that are called salts. So these are skin-associated lymph node tissues. So these guys will recognize microbes that have the potential of slipping past our physical barrier, and then they will take the necessary precautions to essentially destroy them so they don't get past our skin and cause us harm. And then we have Langerhans cells. So these are specifically dendritic cells, and if necessary, they can go around phagocytosing on our skin. But I'm more concerned about y'all knowing why is the skin so important for us. Okay, so with that, we're gonna move down into our mucosa membranes. So this are mucosa membranes. So if you think all the mucosa membranes that we have is our nose, our mouth, and our genital region, primarily for females, not so much for males, just based on anatomy, but they do have um, a mild mucosa area, if you want to say it. But mainly for females, definitely this is more relevant. But these membranes, they are selectively permeable, so meaning that they do allow certain things in or out. But a lot of times um, it is very selective. So sometimes it won't let something out depending on what it is, and a lot of times it won't let anything in. So it is a border. And then also, it does aid in um, not allowing invading pathogens to enter us. And the fact that they are very wet and sticky, essentially, they will trap microbes. And then the fact that they trap them, we end up actually just digesting them. And then they don't cause us any harm. It happens all the time. Like your nose, constantly digesting, as well as your mouth. And then within our mucosa membranes, we do secrete enzymes, and these enzymes are the ones that will actually kill the trapped pathogens if there are any within our mucus. And then we also have something that are called MAMPs. I might mention them as PAMPs, but I'll try my best to say MAMPs. And then we have malts and galts. So malts and galts, they are very similar to salt. The only difference is where their location is. So malt is specific for the mucous membranes, galt is specific for the gut, but they do kind of do the same thing where they will recognize pathogens, and then based on that, it will it'll create the necessary cells in order to take care of the potential threat. And then, actually, guys, I think I said something backwards in class. I don't remember. I think in class I accidentally said, so I'm going to draw a cell. So here's a cell, and then here's another cell. So I think in class I accidentally said that the host has MAMPs, and that is completely incorrect. I'm so sorry, guys, if I did say that in class. So this is a host cell, and then this is the bacteria. So host cells, we have something that are called PRRs. So these are pathogen recognizing receptors. And then bacteria, or like any other microbe, they have the MAMPs. Or I might end up saying PAMPs. They're the same thing. And if I did flip that in class, I am so sorry, guys, for the confusion. But we do not have MAMPs or PAMPs. We have PRRs. We have the receptors for them. I'm so sorry that if I caused any confusion. We'll go more into detail into PRRs later in this lecture, but I just wanted to point that out just in case if I did flip it in class. Okay, so with that, let's go ahead and carry on. We're going to go further into our mucosa membrane. So within our mucosa membrane, further down, like away from like our nasal and our mouth area, we have something called prior patches. So this is actually located inside of our intestine, primarily our small intestine. And it's like they're little tissues, or like little tentacles, and they purposely try to capture microbes. And then within that, we have these M cells. And M cells are a specific part of prior patches. These are in between epithelial cells of our intestines. And what they do is that they act as a filter. And as that filter, if anything needs to be phagocytosed or like chopped up, the M cells will actually do that and disintegrate, digest the microbes if necessary. So let me go ahead and show you what a prior patch is. So this is a prior patch. So this is as if you were like looking down like the tube of your intestine, which is kind of a weird thought. But it is pretty much just a hairy tube, I guess. And then if we zoom in, so this is a prior patch zoomed in, then we have these M cells. These M cells will specifically phagocytose anything that is needed. And then from that, macrophages will come in and help clean up the the mess if there is any pathogens. Okay, so with that, let's go ahead and go to our lungs. So our lungs, like we talked about in our last chapter, it doesn't really have a lot of microbiota in it. So it's not really, it doesn't have a lot of microbial growth, ideally, theoretically speaking. And then the reason why that is thought is because it is highly ciliated, meaning that it has a lot of those tentacles, and in the fact that we have a lot of those cilia, we're capturing a lot of the microbes. And since we're capturing a lot of microbes and then we're expelling them through sneezing, coughing, anything of that nature, mucus production from our lungs, it is thought that our lungs aren't natural harbors for microbes. But if there is any microbes that do end up getting in there, like if somebody sneezes on you and they had like the cold or something, you would ideally be sneezing out to clear out any organisms that were located inside your respiratory tract. And then also, if an organism does make it deep within inside our lungs, so the alveoli, so like where gas exchange occurs, the like the little air sacs in your lungs, it is not easy for us to expel it out at that point by sneezing and coughing. So a lot of times what will happen is that phagocytic cells, primarily macrophages, they will come in and clean up the bacteria if there is any, and then if necessary, they will also send out chemical signals so that we can create an adaptive immune response, as well as aid the macrophages if necessary, if they need help. Okay, so with that, let's go to chemical barriers. So all the ones that we've talked about prior, those are physical barriers. So let's talk about a few chemical barriers. So a chemical barrier is like the enzymes and the pH levels that we have in our body. So we are slightly acidic in certain areas, such as our skin, but primarily in our stomach. Our stomach is very, very lethal to a good chunk of pathogenic bacteria. And that is a fabulous thing because you don't want microbes in your tummy, at least the bad microbes you don't. And then also, we produce a lot of enzymes, primarily such as lysozymes themselves. These are located in a lot of places, but definitely in our tears. And we talked about this in, I believe it was chapter 14, where these tears, these lysozymes in our tears, they degrade gram-positive bacteria. And the fact that they degrade gram-positive bacteria, we won't get eye infections as much, although you can definitely get eye infections. That's perfectly normal. Most of us have probably had an eye infection, but it will most likely not be caused by a gram-positive bacteria. So that's one enzyme type. Another one is lactoperoxidase. This one also does the same thing as lysozyme, where it will go around killing harmful bacteria to us. And it does that by making radicals. I'm not too concerned about you knowing that. I really just want you to know the purpose of the enzyme. So just know that majority of the enzymes that we're going to talk about, their whole purpose is to degrade and kill pathogenic bacteria. And then the last one, it is called defensins. These guys, they are specific to the innate immune system. They're located in a lot of places. They're located in the skin, the lung, the urinogenital area, as well as the gastrointestinal tract. These guys, what they do is that they kill by destroying the cytoplasmic membrane. So what they do is essentially poke holes in the membrane. So the fact that the cytoplasmic membrane integrity is impaired, the bacteria can die. And then it is kind of a broad spectrum cell, if you want to think of it that way, because of the fact that it does provide a defense against gram-positive and gram-negatives, as well as fungal and some viral infection pathogen. So good for defensins. We do love our defensins. Okay, so next we're going to talk about inflammation. So inflammation, the big thing that I want you to know about inflammation is what it is, how does the whole process get started, what happens, and then how do we end it. So let's go ahead and just start like, what is inflammation? So inflammation is I don't want to say side effect, but it is a result of a innate immune response. So the whole process will begin once you have damage to tissue. So once you have damage to a tissue, you will have inflammation, and that will happen whenever macrophages are in the area. They notice that there is a breach in defenses, so breach like in their skin or something, and then from that he will start engulfing microbes, as well as release chemicals which are called chemoattractants, and these guys, they call for help. Okay, and then on top of the macrophages secreting out these chemoattractants as well as cytokines, this will cause vasodilation. So with vasodilation, that's whenever you get like the heat and everything, you have increased blood flow. And then with the vasodilation, it also allows other molecules to travel a little bit more freely between your blood vessel and tissue, which is good because you want your immune cells to get to the area as quickly as possible and as efficiently as possible. Okay, so that's pretty much the exact same slide. Sorry, guys. If you want, I did play the animation in class, but the animation's right here for your convenience, so you can sorry guys, lost my train of thought. So you can review the inflammation process. Okie dokie. So this is just another slide about inflammation. So this is the stepwise process. So you have, like I said, you have the damage to the tissue. You have the macrophages that begin to like engulf all the bacteria and everything, as well as start secreting cytokines and everything to call for help. And then from that, those cytokines will travel to the blood system and everything. From that, the blood vessel will change, and then it will become more permeable. With the fact that it's more permeable, there are other immune molecules that can enter and then can also aid in the whole process. And then from there, the fight will go on. That's essentially the big picture that I want you to get if you're going to get anything from it. But like I said, definitely this animation is fabulous, so definitely take a look at it, please. Okay, but with that, let's go into different types of inflammation. So the main one that I'm going to talk about is chronic inflammation. And then after that, we're going to talk about how do we actually end inflammation. So chronic inflammation, this is where it is a persistent inflammation over time of a foreign body. It will, if it's not resolved, it will cause permanent tissue damage. Even though your body is trying to repair itself, since it's always inflamed, your tissue is going to be damaged to at least some extent. And then there are many causes to chronic inflammation. It doesn't have to actually be microbial based. It can actually be like genetic based, such as like arthritis, like rheumatoid arthritis, that does have a genetic component to it. But there are a lot of microbes that do this, and that's M. tuberculosis. He does that. Also, there's a lot of parasites. And the reason why these guys are very good at causing chronic inflammation is because they can avoid or even actually fight off our immune system. And with them being able to fight a good fight, essentially, they will cause harm to the body, and a lot of times that is a result. Um, Inflammation is the result. But our body does try to maintain inflammation to a specific area as much as possible. And one mechanism is through granulomas. So this is where our body will be like, okay, we need to wall off, we need to contain this inflammation process because of this pathogen, and it will build a wall essentially around the inflammation site so that it is contained. And the only one I can think of off the top of my head that does this is a fungal infection, that's Cryptococcus. But this is whenever the infection is like disseminated. It's very in the late stages. So like somebody has had Cryptococcus for a while, because Cryptococcus, he does have virulence factors of avoiding our immune system. But this will happen a lot of times like in the lungs. So it will essentially create like a lump or like a stone inside the lung. It's kind of like a giant webbed ball that the body will do so that the crypto is contained in that area. So that's the only example that I can think of off the top of my head. Okay. So I think, oh, my slides are a little out of order. I'm sorry, guys. I think I even said that in class. Oh, I'm losing my head. Okay. Anyway, so how do we actually recognize self from non-self? So, like, how do our cells know not to go around just phagocytosing everything? So we do have a self-recognition system, and that occurs whenever there are certain glycoproteins located on our white blood cells. So what will happen is that whenever they're circulating around, it will recognize certain receptors or glycoproteins on each other's cell surfaces, and based on that recognition, it knows that, oh, you're cool, dude, I'm not going to attack you. But as soon as there's a different reaction, a different reception, then the macrophage or a dendritic cell, it'll be like, oh, you're not cool, you're invasive, I'm going to eat you and engulf you. So, but there are ways for bacteria to avoid that. And the main way is actually what it does is that it will create a capsule, as you see right here. So what it does, it will actually protect its cell surface. So meaning that the macrophages, they can't bind to their actual cellular surface, which is that black area. It can't bind to it. So the fact that it doesn't bind to it, it won't be able to recognize that it's self or non-self. So it'll just be like, oh, I just bumped into somebody and carry on. Whereas that if it was like binding to this guy, to this strep guy, then it'll be like, oh, I can recognize you because it doesn't have a capsule. But the 4 up here, it won't be able to recognize it because its cell surface is protected. Okay? Okie dokie. And then I do want you to know the definition of optimization. So optimization is a process where the innate and the adaptive immune system work together. So it's a process that the, like I said, the innate and the adaptive immune system work together in order to facilitate phagocytosis. So meaning that they work together so that there can be optimal phagocytosis of cells that are not good to the host, to us. Okay? Okie dokie. So let's go ahead and move on. So during phagocytosis, there is 2 different pathways that can happen. So that's either the anaerobic or the aerobic pathway. So the anaerobic is where it's oxygen independent, meaning that oxygen is not required. And then we have aerobic, which is where oxygen is required. But the whole process of phagocytosis is to make a phagolysosome. So in order to make a phagolysosome, there has to be fusion. And then depending on whether it's aerobic or anaerobic, doesn't matter, phagocytosis will occur. The whole process will occur where the pathogen is degraded. But depending on if it's a independent or dependent pathway of oxygen, will determine what kind of enzymes are made. So in oxygen independent, you'll make more lysozymes and defensins, whereas oxygen dependent, you'll make more superoxidases, hydrogen peroxide, as well as radicals. So that's really the only difference. But still, the whole goal of phagocytosis is to degrade and form a phagolysosome. But there are some bacteria that can reside intracellularly inside the host cell, meaning that they can live inside of our cells. The main ones I'm going to point them out because we are going to see them again later in term. So that's Shigella, Listerella, and Salmonella. So Shigella, he can actually escape the phagosome, so meaning that he can literally kind of be like I'm not very good at football, like a running back, I think that's what it is, where he's like avoiding the lineman. So that's pretty much what he does, is that he plays a keep away from the phagosome. And as well as Listerella, he does the same thing. And then we have Salmonella. So Salmonella, what he will do is that he will actually attach to the phagosome. And then with him attached to the phagosome, the lysosome cannot attach. So there is no formation of a phagolysosome. So therefore, Salmonella cannot be degraded. And then Shigella, he also can do something else. So what he can do, if he doesn't evade the phagosome, what he'll do is that he'll enter the host cell, and then what he will do, he will kill it. He'll kill the host cell by triggering cell death, and which is coined apoptosis. So apoptosis is cell death, is programmed cell death. And then Coxiella, what he does is that he will produce a toxin, which is bad for the phagolysosome. And since the toxin is being produced, the phagolysosome cannot function and cannot degrade Coxiella. Okay. So, the mechanism for actually surviving phagocytosis, there are 4 main ones. So the first one is that a pathogen will have a capsule. So with the fact that it has a capsule, he will not be phagocytosed and he can't be engulfed because of his MAMPs being protected. And then you have the escape of the phagolysosome, meaning that they literally play keep away. And then you have the prevention of the fusion of the phagosome and the lysosome together. So if that fusion doesn't occur, then the pathogen cannot be degraded. And then you have triggered cell death, where a pathogen can induce suicide of the host cell. Okay, so now let me take like a sidestep, going back into how do we actually end inflammation. So sorry guys, it was a little out of order. But so we had the whole process of where like macrophages, they are promoting cells to come in and vasodilation. But what will happen is that after the infection is all cleared, the neutrophils, what they'll sense that, okay, our job is done, all of our enemies are taken care of, and they will undergo cell death. They will die. And then what will happen is that a macrophage will come in and he will remove the debris. So he'll come in and clean up the battlefield, and inflammation will cease because macrophages as well as other immune cells are no longer being signaled to go to that area. So once that has ceased, neutrophils specifically will undergo cell death, and then macrophages will go in and clean up all the debris, all the carcasses, essentially. Okay. So with that, let's talk about what are other types of molecules in our innate immune system. So we have something called interferons. These are a type of cytokine. We make these in response to intracellular infections. So there are two main types that I want you to know. There's type 1 and type 2. So type 1 is comprised of interferon, which is going to be abbreviated as IFN. There is alpha, beta, and omega. These guys, they function for antiviral infections. So if you have a viral infection, then you'll have increased interferon, alpha, beta, and omega. And then there's type 2. So type 2 is interferon gamma. He's an immunomodulator, meaning that he will regulate our immune system, essentially. He'll regulate the innate immune system by triggering like signaling cascades within other immune cells, such as macrophages. Okie dokie. So, with that, I've kind of already said some of this, but with interferon type 1, so that's the alpha, beta, and omega, these guys specifically, what they do is that they will bind to uninfected cells, and then with that, they will induce production of certain proteins, and that's specifically double-stranded RNA activated endonuclease, as well as protein kinases. So, and then that is required for translation of viral RNA. So, if interferon type 1s, if they are present, then a lot of times you'll have increased production of the double-stranded RNA activated endonucleases as well as protein kinases. And then interferon type 2, these guys, what they do is that they go around activating white blood cells, such as macrophages, killer cells, and then T cells. And then with interferon type 2, so that's interferon gamma, he will go around, he'll increase a certain protein that's called the MHC protein on our cell surfaces. So these proteins are very, very important for presenting bacteria to other cells. So we want to have MHC proteins on our cell surface so we can help fight against bacteria. Okay, so with that, let's talk about what are natural killer cells. So natural killer cells themselves, they kill intracellular infections. So if there's an infection inside the cell, it will go around killing. What they do is that they will recognize that cells have lost their ability to express the MHC1 proteins. So essentially what will happen is that if a what's it called? If a natural killer cell binds to a MHC protein on our cell surface, it will not attack. But if it notices that like our host cell does not have an MHC molecule, the natural killer cell will degrade the cell membrane of the host cell and essentially kill it. So that is what I mean by it will recognize cells that have lost the expression of MHC1 molecules. So let's summarize that second bullet. So natural killer cells. If MHC1 protein is present, the cell will survive. But if natural killer cells recognizes that there is a lack or no MHC proteins, natural killer cell will kill host cell. And it will kill host cell by creating pores inside of the cellular membrane. Okie dokie? Okay. I believe that was the main thing I wanted to mention. I do want to mention that FC receptor because I do believe I mentioned that in class. So on top of them being able to recognize MHC proteins, they also have the FC receptor on their surface. So what will happen is that a natural killer cell will bind to an FC receptor, and then from that, it will be able to link an antibody, essentially. That's really it. I'm not actually too concerned about that. I'm more concerned about you knowing the second bullet point on this slide. Okay, so with that, let's go ahead and go and talk about our MAMPs and our PRRs. So, like I said, we have PRRs on our cell surfaces, and there are MAMPs on the pathogens. So, like bacteria, viruses, fungal parasites, that's what they have. So, we briefly talked about TLRs, I believe, in um, our last lecture, but there's also something called nods. So, NLRs, I'm just going to call them nods because that's a lot easier. These are very, very similar to TLRs, but what they do is that they bind different things. And also their location is different. TLRs are located on our cell surface, whereas NLRs or nods, they're located inside of our cell. So I believe I have a picture later on in the lecture to show you that. But I do just want you to know that they kind of function the same in the fact that they do recognize MAMPs. Now, depending on what MAMP they recognize, will determine the outcome. I'm not going to expect you to know all the outcomes. If I do want you to know one, I will point it out, like one specific one. But just know that a MAMP will bind to a TLR or to an NLR, and from that, you'll have some kind of signaling cascade that will tell the cell that, hey, like we have this infection, you need to do XYZ to get rid of it. Okay? Okie dokie. So I'll go more into nods and TLRs in the next few slides. So this is just a table that briefly summarizes TLRs and nods. This is not a complete table. I believe there's actually, I want to say there's like 14 documented TLRs, and then nods, I want to say there's like 6, if I remember correctly. I can't remember from my microbial class, but I know there's a lot more than this. But from this table, what I want you to recognize is that TLR, like TLR4, he specifically recognizes polysaccharides, and the fact that he recognizes polysaccharides make him a bacteria. He'll recognize bacteria because there are bacteria that do have lipopolysaccharides in their cell membrane. And I want y'all to know, like, go back and look up which type of gram, is it gram positive or gram negative cell that have more lipopolysaccharides in their cellular membrane. So I want you to go back and look that up because that might be a test question, guys. But anyway, so just notice that, like, these guys, they recognize different things. So, like, NOD2 recognizes specifically peptidoglycan. So peptidoglycan is located in gram positive and gram negative, but based on the amount of peptidoglycan, maybe NOD2 has higher affinity, meaning that it prefers binding to a certain gram cell based on the fact that it has more peptidoglycan. So all I want you to know is the receptor, well, each receptor has a preferred MAMP, okay? That's the biggest thing I want you to get from that, okay? Okie dokie. So with that, we're almost done, guys. We only have a few more slides. So this is kind of a nice figure in my opinion. You might even see this again, guys. I want you to be able to dissect this figure. So this figure is specifically showing TLRs on the cell surface, like I said. So this is like our host cell. And then NLRs are located inside the cell. So let's say that we have flagellin located inside, not inside. We have flagellin located with our pathogen, and then TLR5 is the one that is specific for that. So that means TLR5 is going to go around binding to that flagellin, and then once that binding occurs, that attachment, there will be a signaling cascade. I'm not going to expect you to know the signaling cascade. And then from that signaling cascade will go to the nucleus, tell the nucleus to do something, and then a lot of times in this figure, it shows that cytokines are being produced, as well as other factors. There's a lot of other things. It's not just cytokines. But what I want you to know is that you have binding to a specific TLR and MAMP. There's a signaling cascade that travels to the nucleus. from the nucleus uh, will decipher that that message, and then it will have a result, and a lot of times that is production of cytokines as well as other immune factors. So that's the TLR way. Now the NLR way, what will happen is that the pathogen will enter the cell, and then the fact that the pathogen is inside the cell now, it will interact with a nod, and then from that it will interact with a flamosome, and then the flamosome kind of, not really, but sort of acts like a signaling cascade as well. And then the inflammasome will interact with the nucleus, and from that, the nucleus will determine what else is needed to be done. Whether that is to undergo cell death, or if it needs to produce more cytokines, or anything of that nature. So definitely be able to understand this figure and notice at least these maps, okay? Okie dokie. Now we're gonna take a little side step. We're gonna talk about fever, and then we're gonna talk about competency. So fever is actually a good thing. Most people were around 98-ish, 98 and a half degrees Fahrenheit. That is where we like to be. But sometimes we end up having a fever, and that is caused by something called pyrogens. So pyrogens is the actual substance that will cause a fever. And then I just want you to know that. And then also I want you to know what organ actually regulates our temperature, and that's the hypothalamus. So the hypothalamus is our thermoregulator. He is our body's thermostat, essentially. And then also another thing that I want you to know from this slide is what happens whenever we do have a fever. So what happens is that our blood vessels, they will constrict, and the fact that our blood vessels are constricting, we have heat that is building up, and that is where we actually get our fever. And a lot of times whenever you have fever, you have like the chills and the shivers, and then that is also the fact of movement. You're also generating heat, and that's why you get hot. So just think about whenever you've been sick. So the last time you had like the stomach bug, or if you had food poisoning, or even the flu, heaven forbid, hopefully nobody gets the flu or food poisoning, or anything actually. Don't get anything, guys. Stay healthy. But think about the last time you were sick. You had the shivers, and you may even had chills. Then from that, a lot of times you had fever. So it all kind of ties together. But that's all I want you to know for this. I'm not really too concerned about it. I might ask like 1 or 2 questions on it, if that. Okay, so from that, I believe we only have like 2 or 3 more slides, guys. So we have something that's called complement. So complement, this is oh goodness, he is a signaling cascade. That's the best way I can say it. But the whole purpose of complement signaling is to help facilitate phagocytosis and opsonization. So with that, I'm just going to go into the next slide because I feel like it captures complement a little bit better. I do want you to know the 3 pathways. So I do want you to know the classical, alternative, and lectin. And I want you to be able to tell me the differences between them. So the end goal of all 3 pathways, no matter which one, the ultimate goal is to promote inflammation, to promote phagocytosis, as well as opsonization. Now, the differences between them is how they are actually activated. So classical, it is an antibody dependent activation pathway, whereas alternative is not antibody dependent. So an antibody is not present. And then there's lectin. So lectin, this requires the presence of lectin as well as mannose binding site. And that is in response to cytokines being released from macrophages. So let me actually just show you a picture of it. So we have the 3, like I said, we have classical, alternative, and lectin. So these guys, like I said, I'm not too concerned about you knowing every single little step. I really just want you to know the first main step, and then I want you to know the outcome of all 3, which is the same. the same for every single one. The whole purpose is to promote inflammation, phagocytosis, and opsonization. But the difference is their binding. So their binding is that classical requires an antibody, whereas alternative does not require an antibody. And then lectin requires the presence of lectin as well as the mannose binding site. Okay, that's all I want you to know. I'm not too concerned about how it's regulated, so um, just ignore that. Okie dokie. So with that, guys, we can conclude our lecture. I don't know why the slide is located that high, but like I said, that'll conclude our lecture. I know it was a very heavy one. If you have any questions, definitely don't hesitate to contact or ask in class. If not, thank you for sticking around, and I'll see y'all soon.